microbial populations. Got it. So yeah, like you said, I've finished my bachelor's and master's in Boğaziçi University in chemical engineering, and we were working together for my master's thesis, and now I'm doing my PhD in the computer science department. My topic is bioinformatics here in Delft. Here's a quick outline of my presentation. First, I want to start with, well, a short introduction to bioinformatics as much as I can. What we use it for, what it is basically, and then there's going to be a short crash course in molecular biology and also more about the bacterial genomics part of it. And then I'm going to move on to my project that was finally accepted this year. I don't have a link right now, but it's published. It's about using pan-genome graphs to study bacterial populations. Okay. Now, what is bioinformatics? Now that's, well, it's a huge, this interdisciplinary field. With all these advances in DNA sequencing, we've seen that it's much easier and much cheaper to obtain genetic data. And now we have this massive increase in the size of databases that we have. And bioinformatics was more or less born out of this need to interpret and also to understand this enormous data that was gathered by biologists. And this requires tools from computer science. To put it simply and shortly, I'd say our ultimate goal is to understand life. Sure, this is like a cliche, but I think it's still the best and shortest explanation of what we do in bioinformatics. But then that brings the question, what is life? Sure, as everything in science out there, there's no consensus regarding what is life or what the definition is. Although I put a dictionary definition here that you can read. The most widely accepted one would be that organisms that are composed of cells that maintain some sort of an homeostasis, that have a life cycle, and that also reproduce and evolve. Then the fundamental unit of life is cell. It was Robert Hooke, an English architect and a scientist, who discovered that organisms are made up of these cells. In the 1665 book, Microbial, he describes his earliest observations of a microorganism in a microscope. And he came up with the word cell because the plant structure looked a lot like the honeycomb cells. And I thought this was a nice anecdote to put in here. Is there something in the chat? Oh, okay. Okay, so one of the things that I found really fascinating about this whole thing is that despite the apparent diversity in all the living organisms, we're all fundamentally the same inside. The whole biology is like a fight between these two themes, I think. This huge variety in individual in particular, and also the astonishing consistency in the fundamental mechanisms of life. It's estimated that we have around 10 million, perhaps 100 million living species on Earth today, and each species is different. They all produce themselves faithfully, and they produce a progeny that belongs to the same species, and that the parent organism, essentially, they hand down this information that specifies everything about the species in great detail and what the characteristics that the offspring should have. And this phenomenon of heredity, it is essential to the definition of life, and it distinguishes life from other processes that we have, such as growth of a crystal or burning of a candle. In most living organisms, there are single cells. Others, like us, humans, they are more like cities of cells. They are groups of cells that they perform specialized function. They're linked by very sophisticated communication systems. But even for something as complex as a human body, the whole organism, it's been generated by cell divisions from a single cell. So the single cell is, like I said, it's the vehicle for all the hereditary information, and it includes all the machinery to gather raw materials from its environment, and also to construct from them a new cell. And well, again, moving on with the analogy with as a computational system, cells, they store this information. And again, I find it very surprising that all cells store this hereditary information exactly in the same form. So any kind of archival information from one cell, it can be read by information handling mechanism of any other random cell. This hereditary information is stored in the form of double-stranded molecules of DNA. DNA is essentially, it's a long, unbranched paired polymer chain. It's formed always of the same 4 types of monomers. These monomers are chemical compounds that are called nucleotides. Just like sequence of ones and zeros in a binary computer code, this encodes the information in a computer file. And we can take a piece of DNA from a human cell and insert it into a bacteria, and that piece of bacteria cell inserted into a human cell or vice versa. And the information can be successfully read, it can be interpreted and copied. If we get a bit more technical here, the DNA, it's made up of, like I said, simple subunits. These are called nucleotides. Each consists of a sugar phosphate molecule with a nitrogen-containing side group. Or a base attached to it. These bases are 4 types, adenine, guanine, and cytosine, and thymine, that correspond to 4 of the distinct nucleotides, A, G, C, and T, or just like in the movie Gattaca, which is a really great movie. I strongly suggest that you watch it if you haven't seen it already. It's basically a primer for all the bioinformatics students. And moving from that, now DNA, like I said, it copies itself, but in order to carry this information from the parent to the child, it has to do more than just copying itself. It must also express this information. So that means letting this information guide the synthesis of other molecules in the cell. And this expression occurs by a mechanism that is, again, exactly the same in all the living organisms. This mechanism produces 2 of the key polymer types, RNAs and proteins in the cell. It begins with a templated polymerization called transcription. Now, we don't need to go much detail into this, but this is just to give you a rough idea about how this works. But in transcription, different segments of the DNA sequence are used as templates to synthesize shorter molecules of closely related polymers that are called RNAs. Then we have the translation. In translation, these RNA molecules, they direct the synthesis of proteins. In RNA, the backbone is quite similar to DNA, but it has a slightly different sugar. It's a ribose instead of a deoxyribose, which gives it the name RNA. Also, one of the nucleotides, the thymine nucleotide, is replaced with uracil in RNA. And the outcome of this whole process is a polymer molecule with nucleotides that represent a portion of the cell's genetic information, although it's written in a slightly different alphabet. The same segment of a DNA can be used repeatedly to guide the synthesis of many identical RNA molecules. That means the genetic information that is in the form of DNA, it's fixed. But these RNA molecules, these RNA transcripts, they are mass-produced and they are disposable. You can think of them as like a middleman. They just go to the main boss, the DNA, they pick up the information, and then they use this. Well, they are the roots of transfer of this genetic information. And the most important of these are the messenger RNAs. They also called mRNAs. Now, protein molecules, like DNA and RNA, they are again long, unbranched polymer chains. They are formed by gluing together small building blocks of monomers. Again, it's the same idea. The only difference is that these set of monomers, they are different than the nucleotides that we have. Now we have an alphabet of 20 letters instead of 4. We call these letters amino acids. Like DNA and RNA, proteins carry this information in the form of a linear sequence. There's so many different proteins in each cell, and if you discount water, they are the largest portion of a cell's mass. And protein molecules, they're essentially polypeptides. They are created by joining these amino acids in a particular order. And through billions of years of evolution, this sequence, it has been selected to give the protein a useful function. Proteins then fall into a very specific 3D structure with reactive sites on surface. They can bind to other molecules and they can act as enzymes to catalyze reactions within the cell. In this way, proteins direct the vast majority of chemical processes within the cell. Proteins also have other functions as well, so they can be functioning in maintaining the cell structure, generating movements, or sensing signals and whatnot. Each protein molecule performs a specific function according to its own genetically specified sequence of amino acids. And proteins, well, among all, they are the main molecules that they get the stuff done in the cell, basically. The whole DNA, the whole molecule, they are very large, and they contain the specifications for thousands of proteins. But there's special sequences that we have in DNA. They serve as punctuations. Essentially, these sequences define where the information for each protein begins and ends. And each individual segment of the long DNA sequence, they are transcribed into separate mRNA molecules. These mRNA molecules, they each code for different proteins. And each of these DNA segments are what we call a gene. So that was more or less the boring biological background, the real part of it. But you might be wondering now, how does this information actually get from outside of the molecule and then into our laptops? Well, first we need to sequence the DNA. Again, I won't be going much detail here, but essentially what they do is they produce these sequencing machines. They produce small fragments of DNA. We call these reads. And there are 3 different approaches, I'd say, to do this in increasing order of technology, I'd say. The oldest one is Sanger sequencing. This is from 1970s. Then we had next generation sequencing, which basically revolutionized the whole field of bioinformatics because it can generate millions of reads and it is very, very accurate, like 99% accuracy. But the downside is that the read length is pretty short. It's 50 to 250 base pairs. That's when they came up with the 3rd generation sequencing. Now you realize the title of these techniques aren't really that creative, but there are 2 main companies that do the 3rd generation sequencing. That is Pacific Biosciences and Oxford Nanopore. As the name suggests, they are also called the long read sequencing. The reads are longer, almost more than 100,000 base pairs. For example, with this technology, you can sequence viruses directly because they're very small. They're much, much smaller than the read length. But the only downside is that they are a lot less accurate than the next generation sequencing with smaller reads. And once you have all these reads, the next step would be to assemble them. Again, not going into much detail here because this is like a whole PhD thesis here. But basically what you're trying to do is that you're trying to figure out where each piece of this DNA, where each read should go, which ones should be linked to one another. And this diagram here is a nice, simple explanation of what we call a reference-based assembly. In this technique for assembling the genome, you start with a high-quality, complete genome that you already had before, and then you use this as a guide to try to figure out how your new genome should look like, basically. Now, this is where we end the, introduction, mostly, I'd say. So now we can focus more on the specific topic about bacterial populations. But also to give you a bit more context on this, when we think of all the organisms, the whole tree of life, we classify them into 2 groups. We have the eukaryotes and the prokaryotes. The eukaryotes, they are differentiated by the DNA. Their DNA is in a distinct membrane-enclosed intracellular compartment within their cell. It's called nucleus. But prokaryotes, they don't have any of such compartment to house their DNA. Plants, fungi, and animals are eukaryotes. Bacteria and archaea are considered prokaryotes. Most prokaryotic cells, they're small, as the microbial name suggests, and they're quite simple looking, and they live mostly as independent individuals or in loosely organized communities rather than multicellular organisms like humans. They are usually spherical or rod-shaped, and they are very small. They often have a tough protective cell coat that we call a cell wall. Beneath that, there's plasma membrane. It encloses a single cytoplasmic compartment that contains all their genetic information, the DNA, RNA, proteins, and all the other molecules that you need for life. The prokaryotic cells, there's enormous variety. They live in very diverse ecological niches, and they're astonishingly varied in their biochemical capabilities, far more so than the eukaryotic cells. For example, you have the organotrophic species. They can utilize any kind of organic molecule organic molecule as food. They can use sugars, amino acids, hydrocarbons, even methane gas. Then you have these phototrophic species. They can harvest the sunlight as energy, whatnot. And visually, we represent these this species diversity with a phylogenetic tree that I put up here. Each branch on this tree, it means there's something new. It means there's a new species that we've encountered. This is the bacterial tree of life. This is at the level of familia, I think. And again, it's all possible to draw up this tree thanks to the genome analysis, because with genome analysis, we have a simple, direct, and a very powerful way to determine the evolutionary relationships between different genomes. The complete DNA sequence of an organism, it defines its nature with almost perfect precision, and it's very detailed because DNA, it goes through many random changes, and all these changes accumulate over time. And the number of differences between the DNA sequences that we observe between two organisms, they provide us a direct way to measure the evolutionary distance between them, essentially. And this approach, this genome-based approach, it has revealed to us that organisms that we used to just lump together as bacteria before are actually a lot more divergent in their evolutionary origins, as much as a prokaryote is different than a eukaryote. Now, if you look at the bacterial DNA, I'd say the key word here is efficiency. Think of it, when you're small, you have a large ratio of surface area to volume. That means you can maximize your uptake of nutrients across the plasma membrane, and you can boost your reproductive rate. Bacterial cells and also the DNA, they are very efficient. Genes on bacterial DNA, they are packed closely together, and they have very minimal quantities of regulatory DNA. These are DNA that are in between the genes that aren't encoding for any of the genes. On average, we have around 3 million base pairs in a bacterial genome, DNA, and that's roughly corresponds to 1,000 to 6,000 genes. We're quite lucky in that regard because the small genome size means that it's very easy to use DNA sequencing to determine and resolve the complete genome sequences of a lot of bacteria out there. The complete DNA sequence, it reveals both the genes that bacterial organism has and also the genes that it lacks. And when we compare the DNA sequence of different bacterial species, we see that while there's so much diversity, but there's also a lot of conserved genes between different bacteria. These genes are what we call homologous. They are genes with similar nucleotide sequences. We also have paralog and ortholog genes. These are two types of homologs. Orthologs are the same genes between different species, and a paralog would be the same gene, the same similar nucleotide sequence within the same species. So it would be more like a duplication of the same gene. One of the earliest and central concepts of bacterial genetics is the operon. So an operon is a group of co-transcribed and co-regulated genes. Operons, especially those that encode physically interacting proteins, they are much strongly conserved during the evolution of bacterial genomes. There's many studies that have shown that very few of these such operons, they are shaped by a broad range of organisms. And these operons are also, they have the same or similar set of genes, and the genes are on the same order in the genome. We call this order a synteny. And this is a very important point to consider also when I start to talk more about my work. The idea of synteny is very important in bacteria because these large, partially conserved neighborhoods of genes, this synthetic neighborhoods, they have very high predictive value, and they can identify novel functional systems or novel functional regions in the bacteria that we otherwise could not. So it's very important to keep this in mind, I think. And another important aspect of bacterial DNA is horizontal gene transfer. That is when the genes, they are transferred between different species of bacteria instead of being transferred from the parent to the child. And the most striking example of these are the pathogenicity islands. These are, again, the gene clusters that they carry pathogenicity determinants, such as genes that encode various toxins and antimicrobial resistance. Then we have the mobilome. These are sections on bacterial DNA, genetic material that they regularly move within the genome, and they also move between different species through horizontal gene transfer. The mobilome can have bacteriophages, plasmids, transposable elements, and genes that are often associated with these elements. You can think of a bacterial genome in two parts. We have the largest part, that will be the chromosome of the bacteria, and often we see these other smaller bits of DNA, and that's the mobilome. And these bits are also crucial to the main prokaryotic chromosome, and they're not always separate like that. For example, viruses, they and many of the plasmids, they systematically integrate into bacterial chromosomes. This can be a reversible thing, which means the plasmid moves into the chromosome and then it moves out to go off to another species. Or it can be irreversible, and that means the plasmid is domesticated, as in it becomes a part of the chromosome. And these large differences, these large changes in the DNA is what we call a structural variant. It usually includes multiple genes. And on that note, I think it's now appropriate to introduce the central problem of my work, basically the antimicrobial resistance. And I realize this might have been forgotten during Corona because, well, we're more interested in viruses right now, but antimicrobial resistance is a silent, it's silent and a growing threat. It is a global concern. Since penicillin was discovered and first, antimicrobials have revolutionized medicine and how we treat bacterial infections. But you can have all the penicillin in the world and it wouldn't make much of a difference when the organism develops resistance to it. I don't know how familiar you are with Dostoevsky's work, but there's always that person, that character with tuberculosis. And just in a couple chapters later, you see that the person dies. So that's basically what used to happen before we had antibiotics. But if you need any stats from today, we know that antimicrobial resistance affects at least 2 million people in USA each year. And it was linked to over 30,000 deaths in the European Union alone. The multidrug resistant bacteria, these are responsible for 15% of hospital-acquired infections, especially the escape pathogens. They are the highest risk, they have the highest risk of mortality in hospital infections. This is a group of 6 pathogenic bacteria, and as the name suggests, they can escape the most commonly used antibiotics such as penicillin, vancomycin, carbapenem, and more. And this increased resistance, also their clinical significance, is what makes this bacteria very interesting and very important to study. And we really need to understand the underlying mechanisms of resistance. so that we can, well, try to combat this issue. But the spread of resistance traits among bacteria and migration of different bacterial species, can have very complex dynamics. For example, we have intrinsic resistance. That is when a bacterial species, it's naturally resistant to a certain antibiotic or a family of antibiotics, because antibiotics are also a natural thing. This is what bacteria use to fight off other bacteria, essentially. We also have extrinsic resistance, or also called acquired resistance. That means these antibiotics, these resistance genes are usually acquired by plasmids or any other means of horizontal gene transfer. And this in extrinsic resistance actually plays a large role in hospital settings, especially. But there's still a lot more that we don't know about this yet. And that's why in my work, to try to combat some of the issues we had with bacterial DNA, we use pan-genome graphs to study populations. Now, a pan-genome graph, it is a graph. It is not a single genome, but it's a collection of genomes, and it is supposed to represent the whole population of bacteria. It can represent evolutionary conserved regions as well as genomic variants. Nodes can be gene sequences or sequences of varying length. And edges on a graph, they follow the genome sequence. In the context of my work, these nodes, they are always genes. And we have the conserved regions. For example, we have the same gene A in genome 1, 2, and 3. And we lump all these genes within one single node in the graph. So this is a more compact and a more efficient way to represent a whole genome, a whole population, instead of using separate multiple DNA sequences for each of the genome. It's also very intuitive because you can just easily see the variance in a population as these branching paths on a graph. And we can apply a lot of graph techniques from graph theory to analyze these populations. And when I first started, I was looking at the current status, the literature about pan-genome methods, and I realized that there was a lot of methods developed since 2005 when the term was first coined, and they're all for different purposes, all for different organisms. And I wanted to get a bird's eye view of the things and to see the big picture and where the field was heading, basically. So I started this comparative study on multidrug resistant bacteria called Acinetobacter baumannii. It is the A in escape pathogens. And these guys they're notorious for their surviving capabilities. They can survive anywhere, everywhere, soil, water, animals, you name it. And I'd say they're relatively little studied compared to more popular types of bacteria like E. coli, I'd say. And this hardness, and combined with their intrinsic resistance to many antimicrobial resistance agents, this contributes to their fitness, and they are even stronger in the hospital settings. And in the study, I evaluated the state-of-the-art pan-genome construction tools. And my goal was to propose general guidelines and some rules of thumb to help with researchers who may want to incorporate these pan-genomes in their own studies and their analysis, especially for microbial genomes. And by general guidelines, I mean to have a sense to explore what questions each tool might be useful in answering and how they behave, basically. This is, this is like a roadmap to the pan-genome universe. It's essentially the vision, I guess, I had when I first started this project. This is the user guide that I was speaking of. It's like one of those quizzes you have on teenage magazines. I don't know if any of you would remember that, but we used to have these in magazines where you would start from the starting point, and then at each point you would be answering a question, and then at the end you would find out which Backstreet Boy you are. In the same way, I have a starting point, and at each step you answer a question depending on your project, depending on your data set, to come up with one final answer in these black boxes. And unfortunately, I'm not going to go much detail into here, but from this whole diagram, I picked up 5 of the tools that I've highlighted in red here for evaluation. These called, well, the names aren't that important, but these are called Rory, Ptolemy, Pangolin, Pirate, and Panaroo. And I have no idea if I'm pronouncing this correctly, but anyway, the reason why I chose these were because these 5 of the tools, the definition of a pangenome in all of them are consistent. The pangenome is a graph, and the nodes are genes again. And 2, they have very similar methodology, but they are still diverse enough. So there's little small differences that it's still interesting to compare them and see how they fare against one another. But this was by no means a comprehensive study. Of course, since I finished this project, a couple more methods have also popped up, so I couldn't include them here. In short, I'd say these methods all boil down to 3 main steps. First, you find the homolog genes. Those are the genes with similar nucleotides. And then you differentiate between paralogs and orthologs. So you try to figure out what genes are actually conserved between the species and not duplicated within the species. And then finally, you just form your graph by linking nodes, putting edges, where the genes should be adjacent on the actual DNA sequence itself. To construct the pangenome, I found this 2019 study where they compared a bunch of multidrug resistant baumannii strains. All of them were isolated from different sources. They didn't do the sequencing themselves. These were just basically were obtained from public databases. But it was a nice set of curated genomes, so I thought I'd just take it. From these 78 genomes, I removed 8 of them. So we have 70 of them left because some of the assemblies were very old. So these were very old genomes with older techniques. I started with first very basic graph metrics. We have the graph size, which is defined as the number of nodes and the number of edges. You think as you have more nodes and more edges, that means you have a more complex graph, you have a larger graph. I've also put these numbers in context with previous studies. And according to literature, the baumannii as a species, the pangenome should have something around 20,000 genes, which is more or less what all the tools have came up with, which is a good sign. But the exact number of genes in the pangenome should probably depend on the data set you have, because if you have a more diverse data set, then you end up with a larger pangenome size and also vice versa. One thing I noticed here was the most complex graph, I say, the largest graphs. These are also from 2 of the tools that are very strict in their homolog detection. So I think because they are more strict, they have more of these nodes, so they split more of these nodes, and they're basically under clustering things. And that made me a bit curious about the parameter settings. So if I was wondering if I could make these a bit looser or a bit more stricter, and I was wondering how much playing with the parameters would change the results. And I found that, for example, with Ptolemy, when I changed the window size, in Ptolemy, when it's trying to define the homolog genes, it also looks for the neighboring genes. So it uses the synteny information that I was talking about before. So I played around with that window size. And also with some of the other tools, I also played around with little parameters that I don't need to go into detail here. But I've observed that the results can change as much as 15%, which is a lot. It's definitely not little. And that corresponds to like 3,000 nodes in the case of Ptolemy. And for the other one, it's like 2,000 nodes at least. And this is, I think, I've seen only one paper basically that talks about this, how these methods can be sensitive to parameters. But because it's not an extensive evaluation, I just wanted to point it out here in this work that, well, these methods can be quite sensitive, and tuning the parameters properly, depending on the organism, it's not as straightforward as it may sound like. So this is a cautionary note, I'd say. The next thing I looked at was what we call pan-genome partitions. In a pan-genome, you have this whole set of genes. And from these genes, you can find what we call core genes. These are genes that are conserved in more than 99% of your genomes in your population. We have the soft core, that's 95 to 99. You have the shell genes, which is 50 to 95%. And you have the cloud genes. These are the very rare genes that are observed in only less than 15% of your population. So I looked at these partitions with all these tools, and I found them to be quite similar when you look at the core gene content. I also calculated pairwise Jacquard similarity, which ranged from 65 to 0.91. And also the numbers compared to previous studies about the baumannii pan-genome, they're again consistent with previous studies. Oh yeah, then we looked at the cloud genes. This is where I observed most of the variation between the different methods. They were quite different. I think unsurprisingly, the most strict tools were in Ptolemy that gave me the largest graphs. They also had the largest cloud gene content. My guess it's about, again, how they approach the homolog detection. And this made me realize that I think the homolog detection step in these kinds of methods are the most important, and that's essentially what determines your graph size and your graph complexity. So I think if you're more interested in the cloud genes, which are the rare variants in your population that are observed very rarely, it will make a lot more sense to pay more attention to your parameter settings and maybe try a couple different values and see what works for the population itself. And from my previous observations, I decided to come up with a method that I think would be better suited, or at least for my application in this case, because I realized that Panero, it's produced more or less average-sized pan-genomes. I think in the absence of ground truth in biology, this is the most, this has the best balance between over-clustering and under-clustering our genes. But there's also this error correction step in Panero that I found it was disrupting the synteny. So it was breaking down neighborhoods of genes that, and I didn't want this. So I wanted to fix that by indexing the graph again using Ptolemy. And I tried to link these broken paths, these broken gene neighborhoods once again from scratch. And I think this would be better suited to identify these large structural variants, which is what I'm especially interested in here. I demonstrated this structural variant calling on two things with baumannii, both related to carbapenem resistance. One is I've located the context of carbapenem resistance. Carbapenem is a type of antimicrobial. I've located the context of these resistance genes. They are located on transposon. And I've also detected the different plasmid structures from the pan-genome graphs. Both of these are significant in horizontal transmission of carbapenem resistance. The carbapenem resistance in baumannii, it's mainly driven by these enzymes called these carbapenemase hydrolyzing enzymes called oxacylinases, I think, also called oxas in short. This OXA23 gene that you can see here in red arrows, it's carried on these transposon structures, but they can be found on chromosomes and plasmids as well. It's hypothesized that this OXA23 gene, it was mobilized with the help of these insertion sequences, the green arrows that you can see here. And these insertion sequences, they not only help move these genes around the genome, but they also act as a promoter. So it is possible that if you have this OXA23 gene alone, you can have resistance to carbapenems. But if you also have this promoter region, this promoter gene insertion sequence, it acts some sort of a booster. So we see higher levels of resistance. And that's why it's really important to, well, to be able to observe these neighborhoods on the gene, on the genome. Here in this graph, I have extracted a subgraph. So first I found a gene in my graph, and I've extracted a couple nodes upstream and downstream. And I've drawn this small subgraph from my pan-genome. And I've colored the edges to indicate different transposon structures that I've showed you before. For example, you can follow the red path. Here you have, it was called the TN 2006, I think. Yeah, with 2 insertion sequences located upstream and downstream. And then you have the 2008 structure. It's the green path, and it's lacking the second instance of insertion sequence. And now in this case, except for the transposon 2009, I was able to extract the full path of the transposons. But I think with the third transposon, the 2009 one, the path was too long, and it included 4 additional proteins. So I guess it's just I couldn't preserve the synteny, and well, the variant calling wasn't powerful enough to reach that long DNA sequence, I guess. I don't know how else to put it, but that's my idea. And the next, I guess, demonstration I had was with the replicase 1 group of plasmids. So the first part was about transposons. They were smaller, but this one was at the level of plasmids. These plasmids, they're a type of conjugative plasmids. They are specific to the Acinetobacter genus. And they are interesting, again, because they carry around these genes that are associated with antimicrobial resistance and with several beta-lactam antibiotics. And they also serve as a vehicle to transfer these genes. They are characterized by this replication protein called RepB in blue arrows. But you can have different genes that are inserted and removed in solitary variants. At the top, we have the core structure called PA11 because it was observed in a strain called A1 that doesn't have any of the resistance genes. But according to literature, in my data set, I should have at least 3 of these variants. For instance, if you look at the third variant, you see the OXA58 gene in orange, which is a beta-lactam resistance gene. And it was inserted with the help of these green arrows, the insertion sequences. Again, I went back to my pan-genome and I tried to extract these structures from it. I have the edges to indicate different paths of the different structures of plasmids, and I also put the names of these different structures in the legend here. First, we have the green path. It is the core structure. Then we have the purple, blue, and the red paths for the variants. And now for the red path, the variant 3, I call this tricky because in some of the previous studies, I've seen that this variant was mistaken to not contain this plasmid defining rep module because it had a slight mutation. It had a few nucleotide differences. But despite that, in our graph, I've seen that it appears still close to the rest of the variant. It's still located within close proximity to the other variants. And that is because we have this neighborhood information that can help us, even though there's a nucleotide change, we can still keep that intact. And one final interesting thing I've seen was a novel variant. I'm still a little cautious. It's been over a year since I finished this project, well, it's a variant that essentially I couldn't find any information about it in the literature, at least nothing experimental. I don't know how it relates to other plasmids exactly, but to my knowledge, it hasn't been studied extensively yet, but it looks like a new structure with even different resistance genes compared to the other ones. So it would be a very interesting thing to study, I think, and sequence more genomes to see if we can we can find this in the other ones. Yep. So to wrap up, in the study, well, we first developed a guide, a general guide, like a roadmap for researchers to the pan-genome universe for those who want to use pan-genome graphs when they want to study microbial organisms, especially. I've also done a small-scale evaluation of the state of the art. I compared graph sizes, different pan-genome partitions. And I've seen that while methods were more or less consistent with the core content, they were most different in their cloud genes. So if you're more interested in the rare variants on your population, it would make a lot more sense to be a bit more careful about the tool that you're choosing, as well as the parameter settings that you're trying. So there's a lot to gain by not using those tools just straight out of the box. In the second part, I've combined two of the tools that I was evaluating so that I can come up with something better, something more suited for structural variant calling. And we demonstrated this effectiveness on two things, to call structural variants on transposons and plasmids that are associated with carbapenem resistance in baumannii. I've seen that while it was very well suited to identify structural variants at small DNA sequence at small distances in the DNA, as the distance became larger, it also became a lot more difficult to locate and also extract these variants. So it looks like there's a lot more work to be done there. I've also, well, again, being cautious, but I think I might have identified a novel plasmid structure here that is waiting still after a year and a half, still waiting to be studied. But overall, I think this project was provided really valuable insight into both the biology and also the technical side of pan-genome grafts and multidrug resistant baumannii populations. Yeah, and to that note, I'd like to end my presentation here, and I want to thank the Delft Bioinformatics Laboratory and also my supervisor, Thomas Abil, for his help. Yeah, if you have any other questions, I'd be glad to hear them. Although we don't have much time left now, I think.